DNA ligase is a very important enzyme and it has different types of functions. It works on replication, repair mechanism, recombinant DNA technology, and also in the cloning purpose. Now we have to understand that how DNA ligase works. So we have to understand the mechanism of the enzyme DNA ligase. So there are main three steps through which we can easily understand the mechanism of DNA ligase. So first, that is addition of AMP. Second step, that is activation of the 5' phosphate region of the DNA ligase, and third, that is removal of AMP, and the gap is sealed. So the first step, that is addition of AMP to the enzyme. So AMP from ATP or NAD plus, nicotinamide adenine dinucleotide phosphate, is attached to the lysine residue present on the active site of the DNA. Active site of the enzyme. So the AMP is transferred from ATP or NAD plus to the amino group of the lysine residues. Now, so first step, that is the transfer of the AMP to the amino group of the lysine residue. Now the next step, that is activation of the 5' phosphate present in the Nic region of the DNA. So the enzyme AMP complex is formed in the first process because AMP is transferred to the amino group and enzyme AMP complex is formed. Now this AMP is transferred to the 5' phosphate region of the DNA present in the Nic region of the DNA, and it leads to activation of that particular phosphate. So second step, activations of the 5' phosphate, and during this process the 3' OH attacks this 5' phosphate. This is called as a nucleophilic attack. So the third step, that is removal of AMP. So 3' OH attack the 5' phosphate and the AMP is removed, and ultimately the gap is sealed. So, and phosphodiester bond is formed. So what is the purpose of the DNA ligase? DNA ligase, the main purpose of the DNA ligase is the formation of phosphodiester bond between 3' and 5' end of the DNA. So now there are different types of DNA ligases, like Ecoli DNA Ligase. Mammals have different types of DNA ligases. Like T4 bacteriophase has different ligases. So the main purpose of the DNA ligase is same, but the requirement of ATP is different from one organism to another organism. Like in case of E. coli, the DNA ligase uses ATP as an energy, NAD plus as an energy source, means nicotinamide adenine dinucleotide phosphate as an energy source in the formation of phosphodiester bond. Next, that is T4 bacteriophage or T4 ligase, that is isolated from T4 bacteriophage and also eukaryotes, uses ATP as an energy source in the formation of phosphodiester bond. Mammals have different types of DNA ligases, like DNA ligase 1, DNA ligase 3 and DNA ligase 4. So DNA ligase 1 works on the lagging strand. Ribonuclease H remove the RNA primer from the eukaryotic fragment, and ultimately DNA ligase works on that particular area and the gap is sealed. Now DNA ligase 3 complex with XRCC1 and form a complex and works on the nucleotide excision repair mechanism. DNA ligase 4 also complex with XRCC4 and form a complex, and it works on non-homologous end joining, that is a double-stranded break repair mechanism, and also BTJ recombinations. Now we see, let's see on the paper the details mechanism of the DNA ligase. So DNA ligase is a very important enzyme. DNA ligase is a very important enzyme, works on replication, DNA repair mechanism, recombinant DNA technology, and also in the cloning purpose. So replication, DNA repair, recombinant DNA technology. DNA ligase works play a very important role in case of replication, DNA repair, recombinant DNA technology, and cloning purpose. So mechanism of DNA ligase. So mechanism of DNA ligase. So this is the structure. So this is our DNA ligase. DNA ligase, the active region contains lysine residue, and this is the amino group of the lysine residue present on the active site of the enzyme. So this is our DNA ligase, and the active site contains lysine residue. So AMP from ATP or NAD plus, nicotinamide adenine dinucleotide phosphate. So AMP is transferred from ATP or NAD plus. This is the structure of the AMP are phosphate ribose and adenine. So this is the structure AMP is transferred from. R means PPI in case of ATP, pyrophosphate in case of NAD plus. R is NMN. Now the enzyme AMP complex is formed. So this is the DNA ligase enzyme, and active site contain NH2 plus NH3 plus, that is AMP is attached, phosphate, then this is the sugar residue ribose and adenine. So this is the enzyme AMP. Enzyme AMP is attached with the enzyme and form enzyme AMP complex. So this is enzyme AMP. Now, so this is the first step of the mechanism of the DNA ligase, called addition of AMP. Addition of AMP to the DNA ligase and form enzyme AMP complex. Now second step is activation of 5' phosphate present in the nick region. Activation of the 5' phosphate present in the nick region of the DNA. Now this is the structure of the DNA: 3', 5', 3, 5', 3', 5', 3'. So there is a 3' OH and 5' phosphate is present in that particular nick region of the DNA. These are the bases of the DNA. Now, so first step is activation of 5' phosphate. So this is the 5' phosphate. Now enzyme AMP transfer the AMP group to the 5' phosphate. So this is the G region: 3', 5', 5', 3' OH, 3'. Now OH region, and this is the 5' phosphate. So AMP is transferred to the 5' phosphate in the nick region of the DNA. This is the phosphate, and this is the ribose sugar and adenine. So this is the AMP attached with the 5' phosphate in the nick region of the DNA. Now 3' OH attack the phosphate. This is the type of nucleophilic attack, and AMP is removed. So 3' OH attack the 5' phosphate. These are the bases. Now third step. So this is the third step. In this step, the AMP is released out and the gap is sealed. So phosphodiester bond is formed in the third step of the mechanism. So this is the phosphodiester bond is formed. Sealed DNA. So this is the sealed bases of the DNA and is called sealed DNA. So in the DNA ligase, mechanism of the DNA ligase, how NH3 group, addition of AMP to the NH3 group, and ultimately enzyme AMP complex is formed. Now enzyme AMP transfer the AMP to the 5' phosphate region of the nick region of the DNA. Now this AMP is transferred to the phosphate. So in the third step, the AMP is released out, and in the first step, the enzyme NH3 is formed because the AMP is transferred to the 5' phosphate region. 5' phosphate region, and this region ultimately AMP is released from the DNA and ultimately